Okay, so I researched the effect of D-limonene from blood oranges on the inhibition of VEGF and angiogenesis in candidiasis elegans. So you're probably wondering what that is, and I'm going to explain it to you. So, angiogenesis. Angiogenesis is the formation of new blood vessels from pre-existing blood vessels. It plays a major role in tumor growth, in tumor development, and it leads to metastasis. So, as blood goes into the tumor expands. And as the tumor expands even more, the tumor expands and expands and expands and develops. And as that happens, it gets too big. And parts of the tumor begins to break off into other parts of the body, which leads to metastasis. And when metastasis occurs, the survival rate in these types of tumors decrease greatly. So, the VEGF. It's a protein. It's the most potent factor in angiogenesis. Its overexpression leads to angiogenesis. So, the process of this is that VEGF binds to nearby endothelial cells. And when endothelial cells divide, they form new blood vessels, which leads to angiogenesis. So as you can see in this picture here, as this tumor is secreting VEGF, it's leading on to attaching to more blood vessels, and as that happens, it's growing in size. So, to inhibit VEGF, we will decrease tumor size and essentially starve the tumor. So solid tumors require an increased blood supply if they continue to develop. VEGF overexpressing tumors can develop this enhanced blood supply by themselves. So, as seen here, as the blood supply goes to the tumor, it expands, and as the VEGF is inhibited, it starves the tumor is dying. So, previous research indicates that the inhibition of VEGF ceases tumor growth. So, you might be wondering, what tumors actually express VEGF? So, pancreatic cancer, glioblastoma, lung cancer, breast cancer. In pancreatic cancer, the survival rate is 2% in advanced stages. In glioblastoma, which is the most common malignant brain tumor, the average survival time of a patient is less than a year, even with therapy. So, in glioblastoma especially, the grade of the tumor, or the rate at which it has expressed, or the rate at which it has advanced, is graded in terms of how much VEGF is expressed in the tumor. So, for example, a low-grade glioma would not express as much VEGF as glioblastoma multiforme. So, I use D-limonene to try to inhibit VEGF. It's a major ingredient in several citrus oils. It's found in the rind of blood oranges, lemons, limes. And it is still under investigation whether D-limonene has an inhibiting behavior on angiogenesis. So, I use C. elegans in my experiment. I use the PVF1 strain, which is a VEGF model, and I use the N2 strain. They are cost efficient, they are easy to maintain, they reproduce quickly and by themselves. But most importantly, they share the same cellular mechanisms and genes as humans. And also, this is a microscopic roundworm. They are supposed to be transparent as seen in this picture. However, I used the PVF1 strain, which overexpresses VEGF. These, as you can see, are not transparent. That is because they are overexpressing VEGF. They have tumors all throughout their body. So, because C. elegans don't have vascularization, the PVF1 strain was used. It's a VEGF model in C. elegans, and it is used to test the VEGF pathway. It contains the same biochemical properties as the VEGF. As seen in the picture, expressing a lot of tumors, and hopefully to inhibit those tumors and decrease them in size, I'm going to use D-limonene. So, current treatment for VEGF overexpressing tumors. There's aggressive surgery, radiation, chemotherapy, and oftentimes, even these don't help. Chemotherapy leaves the patient with really horrible side effects, hair loss, long-term memory, what they call its chemo brain. And most of the time, even with these treatments, the survival rate is very low. So I predicted that D-limonene would be an effective potential chemopreventive treatment for VEGF-secreting tumors. And this is due to previous research on angiogenesis and VEGF, and its ability to inhibit these two things. It also has a low toxicity. It's a natural supplement rather than a toxic substance. And my other prediction was that all N2 test groups, which showed no specific abnormality, would secrete the same amount of protein in the end as the PDF1 ligand would. So to do this, I needed to figure out different sorts of D-limonene concentrations. So I did this through a proportion setup through stearic acid. Because there's not very much research on D-limonene, I had to figure out the D-limonene LD50, which is the toxic amount of supplement that a C. elegans can have, or an organism can have. And I figured out that it was 25.5 microliters per milliliter. So I set up a series of concentrations that the C. elegans would be administered. So the two strains were used, and they were seeded or fed for 48 hours with this D-limonene. And so there was 2 plates for every concentration. So I had 5 different concentrations ranging from 0% to 50%. The 0% D-limonene was the control group. That means that it was fed with no D-limonene. In order to determine the PDF1 protein expressed in the sample, and if it was, and if so, to quantify it, I did an indirect ELISA. So that's basically a 2-step binding process with a primary and a secondary antibody. It's just used to detect the protein. So this is just a series of steps that is included for an indirect ELISA. So results of this test was that in the 0% PDF1 ligand, which had no concentration of D-limonene administered to them, they had a 0.17 protein absorbance rate. That means that that is like the control group. That is the amount of VEGF expressed in C. elegans with no treatment. However, when these same C. elegans were treated with 50% concentration of D-limonene, they had a 300% reduction in tumor size. So, as you can see in these results, the 50% concentration of PDF1 had almost, basically the same amount of VEGF expression as normal C. elegans. So, C. elegans that were not previously treated with D-limonene, or that were not previously expressing VEGF. So, just to remind you of my hypothesis, D-limonene would be an effective potential chemopreventive treatment for the inhibition of the expression of VEGF. And I found that in a dose-dependent manner. As the concentrations of D-limonene increased, the protein absorbance decreased, showing an inhibition of the expression of VEGF. In the N2 worms, which were worms not otherwise having an abnormality at the beginning, I predicted that all N2 test groups would secrete the same amount of protein. As seen here, there was a little bit of fluctuation in the protein absorbance rates, but it really did not have, it was really a negligible effect on protein absorbance rates. So my conclusions. I concluded that D-limonene is associated with a reduction in VEGF expression in a dose-dependent manner in PDF1 C. elegans. D-limonene had a negligible effect on N2 worm samples, and so that was good. This means that VEGF was suppressed with D-limonene treatment. This means that D-limonene is a potential effective natural treatment to suppress VEGF inhibition. And I would also like to thank the Artificial Girls for funding my experiment. Ms. Shofi, Ms. Singh-Sakin, and Mr. Vernon, and Sarah Bohm, who conducted my research with me. So I'm going to open this up to questions. Yeah.